Hello everybody, and welcome to today's webcast entitled Meat Authentication Using LC-MS, the Identification and Detection of Biomarker Peptides specifically for horse or pork in halal beef. I'm really pleased to welcome our speaker today, Dr. Jens Brockmeyer from the University of Munster in Germany, where Jens is currently the research group leader of the food proteomics team within the Institute of Food Chemistry. Jens and his team recently published the first rapid and sensitive mass spectrometry method for the detection of horse and pork markers using MRM and MRM cubed, and he will give us an overview today in his presentation. But before we get to Jens' presentation, I'm also pleased to welcome Locke, the Technical Applications Manager from AB Sci-X, who will first give us a brief introduction of some of the basics of using mass spectrometry for meat speciation. During the presentations, you will have an opportunity to ask a question in the question box next to the slide presentation on your screen. If you have any questions for Steve or Jens, you can write those into the questions box, and we will have a question and answer session at the end of the presentation. Also, you can download a few reference materials highlighting the details of the work presented from the box at the bottom of your screen. This includes the paper that was published in the Journal of Agricultural Food Chemistry and also a couple of AB Sci-X application notes specifically relating to speciation work. Finally, during the presentation, you can also maximize the slideshow display on your screen by clicking the full screen button at the top right of the presentation. So, no further ado, I'll now hand over to Steve to get us all started. Steve, over to you, sir.
Thanks, Ash. Good morning, good afternoon, everybody. I'm going to kick off today's webinar with a brief introduction into meat speciation by LC-MS. It'll be an introduction to Jens's talk, and we'll go through what AB Sci-X have been doing in this area and what we've published and produced over the last couple of years. So the first slide really is just an overview of what we do when we do allergen and meat speciation detection. Basically, we're detecting the protein as a marker of the species. In this way, what we do is we first of all digest the protein. We typically use enzymes like trypsin to break the protein up into peptide fragments. The peptide fragments are then detected by their parent mass in mass spec, and then fragmented in the gas cell to produce a fingerprint MSMS spectrum. From this, we can generate a series of MRMs which can be used to detect these peptides. These peptides have to be chosen very carefully. You have to make sure that they're species specific, so you don't get any false positives. You have to make sure you choose the most sensitive peptides, so to hopefully prevent you producing false negatives. And also you have to make sure you try and choose peptide fragments which are not modified by processing. I know Jens will go into this in more detail in his presentation. So this workflow is very similar to what you used to do or you still do in academia and in proteomics research. So this is the traditional workflow that we have employed to detect species, be it meat or allergens. So one of the first areas we had to overcome was looking at the sample prep. This current slide shows you the traditional sample prep, which we started off in 2009, which basically went through extraction of your food to get your proteins in solution. You then centrifuge to drop out solution the solid material to get just your liquid layer containing your proteins. And then you had to reduce, alkylate, and digest the proteins. This is probably the longest step in the whole process of generating the sample ready for LC-MS analysis. In the traditional way, this typically took an overnight digestion after reduction and alkylation. Reduction and alkylation was used to open up the proteins for the digestion enzyme. In all cases, most people tend to use, Jens and I have used trypsin as the digestion enzyme. Finally, once the sample has been digested, you filter it. Then you clean the sample up by SPE. This removes salts and also concentrates up to the peptides before you blow down, reconstitute, and analyze by LC-MS. So if you look at this workflow, typically when we started off, this would be an overnight analysis, so it would take 20 hours. And so this is based on a paper by Carrera et al. In 2008. So this is the first area at AB Sites we looked at in order to sort of streamline the whole process and ideally get it down to 1 day. And this is what we did in the last couple of years. We majorly looked at the digestion and alkylation step, and what we did was looked at other industries, for example, the biologics pharma industry, which tend to use very quick digestions to speed up the sample throughput. And this is what we did here. We used very fast digestion techniques of, for example, pulse microwave digestion, or in majority of cases, thermal mixing to speed up digestion. So no longer did it take overnight. In most cases, you could do this now in 1 hour. The alkylation digestion step tends to remain the same. And then most of the protocol is very similar. And one step you can also automate is the SPE step. But I think in both in Jens and in my case, we actually did this offline so that we could reconstitute to a low volume to enable use of other techniques to improve sensitivity. But all these changes actually reduced the time of analysis down for an overnight run to 6 to 8 hours. So the goal was achieved in enable us to run a sample in the same day. So mentioning moving to different types of LC approaches, we also looked at modifying and choosing different forms of LC to speed up both the throughput and improve sensitivity. Again, going back to the academics area, typically in this case, nano-LC is used to detect proteins and peptides, mainly peptides in idea of proteins in samples. Nano-LC, however, is very long in run times and has robustness issues because it's very difficult often to connect the LC column to the spray emitter. And one thing which is really good and came out recently was micro-LC. This sort of sits halfway between nano-LC and high flow, but it actually simplifies the whole connection, and it gives you huge sensitivity increases compared to high flow. Basically, you're using the same source as you would use in high flow separation. All you're doing here is changing the probe and the electrode within the probe to reduce the dead volume of the system. This is to prevent peak dispersion and broaden in peak and to improve the sensitivity. So this micro-LC system was recently released about a year and a half ago by Exigent, and it's designed to work at sort of flow rates between 5 to typically 50 microliters per minute. This is where we typically use in our separation, James and I, but you can go up to 200 microliters. And you're using 0.5 millimeter columns typically. The sort of injection volume we use, or I've used in this area, is 10 microliters. So the injection volume itself is similar to what you would use on a high flow system. However, what you do get, as I mentioned earlier, is gains in sensitivity. And here you can see an example of peptide analysis. These are allergen peptides, but the same results you can get with meat speciation. And I know that Jens will cover some examples of this in his talk. But this is what you can typically get when you move from high flow to micro-LC. So here you can see gains of up to 13-fold for one peptide, but typically from 4 or 5-fold gains in sensitivity, moving from high flow to micro-LC flow. And also you have the capability of speeding up the analysis as well, which is dramatically faster than compared to nano-LC. So micro-LC really suits this sort of workflow for allergen and meat speciation. So once we moved our methods over to micro-LC and used the sample throughput, we then applied these two approaches to the area of meat speciation. And this is really driven by the horse meat scandal, which happened about February time last year, which had a huge impact to the food industry and drove us to develop one of the first methods in this area. In this case, what we did is just look at horse. We bought a horse protein from Sigma Aldrich, which is a myoglobin protein, and then we developed our peptides, which are specific for the horse, and then we screened some samples from LGC. LGC provides, are a company in the UK which provides samples for ring trial and performance testing programs for food testing labs. So these were reference material which had beef containing a known amount of horse. And the idea here was for us to develop a method down to 1% horse in beef. And we achieved that, and we actually managed to pick up 3 different markers of horse in beef, and these are specific to the horse and weren't available in the beef. But one of the beauties about LCMS is that you can combine this with also detecting vet residues in the same approach. And this is really important for horse meat. One of the reasons is that horses can contain a drug called bute, which is phenylbutazone. Now, most of the traditional methods used to detect horse meat by LCMS were just based on bute. The reason why they did this is bute is banned from human consumption. It's a drug which is used as an analgesic for horses, so it's used in horses which are used for horse riding and jumping. But it's banned from human consumption because of the negative effects it gives to humans. So it shouldn't be present in horse meat, and therefore it shouldn't be present in beef as well by default. But what we did here was combine the LCMS method for bute with the horse meat speciation method with detecting the protein markers, all in the same sample prep and all the same LC method. So that enabled us not only to detect the horse meat in beef, but also to detect the presence of bute, which was spiked into the sample at the beginning of the analysis, all in one run. So I just want to finish off with some brief conclusions to this short presentation before Jens kicks off his talk. We feel at AB Science that LCMS has a real potential in meat speciation. One of the benefits of LCMS is the multiple points of confirmation, and we have shown in this short presentation that we're able to detect multiple markers for horse meat, as well as the vet residues present in horse in the same analysis. And this is something you simply can't do currently with other methods, for example, ELISA or PCR, because these methods are not suitable or simply can't detect the bute together with the horse meat markers. In PCR, it's DNA, and ELISA, just the proteins. And in the same method, we managed to achieve sensitivities which are at the level of current ELISA and PCR testing and legislation. So we believe we have a method now which is capable of detecting horse meat from the protein markers as well as the banned steroid, so the banned analgesic drug bute in the same analysis. But one of the next areas that we were going to look at, is what Jens has looked at, is what about actually multiplexing species analysis. So not only screen for one variety, but screen for multiple varieties, single and run. And this is something that LCMS is capable of doing, because all you need to do is increase the peptide list to include the other species. And this is an area that Jens has been working on and the University of Munster, and that's the area that he will talk about next. And now I'll pass over to Ash for the introduction of the next talk. Thank you very much.
Thank you, Steve. Nice presentation. It sets the scene for Jens's presentation. What I'd like to ask the listeners to the WebEx today is we have some polling questions that we'd like to ask, and hopefully you can take the time to answer these. Basically, are you currently doing meat speciation testing in your laboratory? So we get some idea of what we're trying to see from here. And there are certainly some interesting results here. 40% of people seem to be showing that they are, where 60% aren't. If yes, which methodologies are you using for meat speciation testing? PCR, ELISA, SDS Page, CE, mass spec, or other? And it seems that 75% of people are using ELISA, or certainly a high proportion as we see the results come in. So yeah, a high proportion of people are using ELISA for doing the testing. Nobody using mass spectrometry at the moment. And what limitations do you experience in your current meat speciation methods? Specificity, sensitivity, throughput, cost, etc. And it seems as though cost is a high proportion, but also we see that specificity and sensitivity, whereby we may get too many false positives or false negatives, are causing the problem. So good interesting feedback from the audience today. Just like to remind you about asking questions before we presentation. The question is to the left-hand side of the display. So if you have any specific burning questions, we can answer at the end. Please do type in those questions and we'll get to them. But without any further ado, I'd like now to turn over to Jens on his research. So Jens, please sir, where you go.
Yeah, thank you very much for the introduction and welcome to everybody. And I'm kind of quite pleased to see that the results of the poll really somehow fits my expectation and what I think about what is the current methodology and so on and so forth. So at least this fits with yeah, some of the slides I will present you in the coming minutes. I'd like to go a little into detail on meat classification using mass spec, and today's agenda. I want to first give a little bit of background and discuss the current methodology and also the limits and limitations of the current methodology. The next step is then the chapter Know Your Enemy. First thing you have to do when you want to do mass spec of biomarker peptides for meat authentication is to discover these biomarker peptides. And I want to go briefly into this. The next step, if you already have discovered and identified your specific peptides, is then the targeted detection of peptides. And I will discuss some aspects of method development, mainly then MRM, detection optimization, and so forth. I would like then to give some words on the improvements in methodology, because ABSIX has done a really good job on development of MRM Cube and the micro-LC. Steve has already mentioned briefly, and I would like to show some data how these two methodological improvements can help the detection. And I'd like to close with some future perspectives. So where will the field move in the near future from my point of view? In 2013, we had really a massive problem with authenticity of food in Europe, and I think that most of you are aware of it and maybe have heard of it. It was the horse meat scandal, where really 10,000s of tons of food contained undeclared amounts of horse and also pork. And this was, of course, a major problem for food industry and also for consumers with religious or ethical concerns against the consumption of horse or pork. So this was really a massive scandal and a massive problem last year in Europe. But if you have a look at recent scandals, you will also see that a couple of weeks ago, fox meat was discovered in China. So obviously food fraud is a problem on global scale. And not only a problem which you observe in Europe. Which methods are currently used? And I'm quite happy to see that this slide fits with the results of the poll. So I think that the main methods that I use are PCR and ELISA. I've seen that some of you also do SDS-PAGE. I would like to go briefly into PCR and ELISA. And of course, you can perform specific and also sensitive analysis with these two methods, but you can see problems with PCR, especially when it comes to the analysis of processed samples. For instance, due to DNR sharing, you can maybe see false negative results, so a loss of sensitivity, which finally then also leads to lack of specificity because you have some false negative results. In general, it is possible using PCR to do some multiplexing, so you can have 3 to 4 targets in one row, but multiplexing is not really great and not really high when using PCR. For ELISA, you have similar problems when it comes to the detection of processed samples. When you imagine that you have your antibody which binds to a tertiary structure of your protein, and you have a denatured protein. This might lead to problems, and in addition, if you have highly and closely related species that you need to distinguish, for instance, donkey or horse, or tree nut allergens are examples. There you might see effects that specificity is not great. Multiplexing in general in ELISA is not used, or when you do multiplex ELISAs, they are in general quite costly. And if you take all these, we think that mass spectrometry really has great potential in species identification, because you can really achieve great specificity, because you really use several MRMs, and together with MRM Cube, I want to go into detail later concerning this novel MRM, which you can also use to increase specificity. You have really the great advantage that you have several MRMs, and you can also use several biomarker peptides per species, and this is really an advantage compared to PCR and ELISA, where you in general only have one target per species, for instance, for PCR. Sensitivity at the moment is comparable with the most sensitive PCR and ELISA methods, so it's already pretty good, and we will see where we will end up with sensitivity. And one of the massive advantages is really the possibility of multiplexing. So you can really have several biomarker peptides per species, and you can then also analyze several species in one run. This is really one of the great advantages for mass spectrometry. When MS is used for authentication, you need to take several aspects into account, and this is given on this slide. The species identifications in general performed by the detection of polymorphisms that are species-specific. However, you will not directly detect the intact proteins that have these polymorphisms, because the chromatographic separation of complex protein mixtures in general is not great. We have to admit, quite poor. So therefore you digest your protein mixture to the tryptic peptides, which then can be effectively separated on standard LC columns. In general, there are 18 columns used, and you then can detect your peptides that cover the polymorphisms. An important point is then the validation of your specificity. So you really have to make sure that your biomarker peptides are species-specific, and that all of your different breeds of your species really show this variance, so that you have really species specificity and not breed specificity. And if you have done all this, the next step is then the optimization of sensitivity, and this is also one thing I want to go into detail later on. So for the first step, the identification and discovery of your analytes, there are in principle two possibilities, and the first one is the in silico approach. You perform a database search where you can have a look for the polymorphisms and choose the appropriate peptides, and then based on these data develop your method. And the second approach is the experimental identification by proteomics experiments, and I'd like to go into detail on both approaches. First, on the database search, the simplest approach is to perform a database search using, for instance, a BLAST website, which I think many of you might know, the basic local alignment search tool from the NCBI. And with this alignment search tool, you can compare sequences of homologous and also of unrelated proteins, and then have a look if you find polymorphisms which you can use. This is exemplified here. I have chosen the hemoglobin subunit alpha from Bos Taurus. Bos Taurus is cattle. And you then perform the blast search to find the homologous proteins. You will end up with a long list of hemoglobins from a related mammalian species. And if you want, for instance, to develop a method to distinguish beef and horse, you will see this sequence alignment from the hemoglobin subunit, which compares the sequence from horse, which is Equus caballus, and beef, Bos Taurus, and you then see the polymorphisms. And here, given in the red frame, you see a potential triptic peptide, which is a potential candidate for your analytical approach. You do see that you have two polymorphisms in this peptide. Alanine and glutamine is exchanged to two serines in the horse hemoglobin. And you can, in principle, use this information to choose this peptide as a potential biomarker and then develop your method. Nevertheless, what is really needed is that you check this peptide again in your BLAST database against unrelated proteins, because it can be that this peptide also occurs in unrelated proteins, for instance from wheat or from organisms and so on and so forth. So this is really needed. In addition, I really strongly recommend to validate the specificity of your peptides using mass specs. Really can be sure that this peptide is specific and only specific for your species you want to identify. However, database search has some limitations. You cannot, at least as far as I can see, you cannot do proteome-wide queries. So you cannot ask questions to the BLAST databases, show me all species-specific peptides of horse. You have to do it protein per protein, and this can be laborious. And the second problem is that the flyability, so the behavior in mass spec of the peptide is not known. It's a complete in silico approach. No idea whether your peptides nicely ionize, whether they fragment nicely, or whether they may bear any modifications. And in many cases, that you have no idea whether the abundance of this protein is high or so. For proteins like hemoglobin or myoglobin, you in general know that they're quite abundant, but for more exotic proteins, this might be a problem. And the next point is that databases in general are not complete. So you have the risk that you have false positive hits, which what might be a problem in the method development. That's the reason why we, for our study, used the proteomics approach. So we experimentally identified our peptides. When you want to do these proteomics approach, you have, however, to keep in mind that the proteome in mammalian tissues is really complex. It is estimated that about 20,000 proteins are in mammalian cells. This leads to about 4 million triptic peptides. And in addition, you see incredible differences in the copy numbers from the proteins in mammalian cells, from 10 to 50 copies per cell to up to 100 million copies per cell. And this is often illustrated by this iceberg shown on the left side. And at the moment, using proteomics, we are able to detect this tip of the iceberg, so we can detect and identify the highly abundant proteins. But in general, it's quite hard to see the proteins with low abundance. For our approach, it's, however, not really a problem, because we anyway want to detect biomarkers from the highly abundant proteins, because we want to have a really sensitive method. So what we are actually doing is With our proteomics approach, we scratch at the tip of the iceberg, so to say, to get as much specific biomarker peptide as possible. And we are able, if you have a really decent proteomics method and a really decent machine, you can identify in one run 30,000, maybe up to 50,000 peptides in one run. So this is in general possible. Oops, sorry for that. And the next point is there are some nice things about the composition of meat. So meat contains up to 21% of total protein. So we are lucky in this situation that we have loads of potential biomarkers in here. And we, in addition, have two highly abundant protein fraction. This is on the one hand the myofibrillar fraction, which is about 60% of the total protein, and this myofibrillar fraction contains all the proteins from muscle fibers, and the sarcoplasmic fraction is composed mainly of the highly abundant enzymes. The connective tissue is quite insoluble and contains mainly collagen and elastin. So we went mainly therefore to extract the highly abundant proteins from the myofibrillar and the sarcoplasmic fraction. And that was the reason why we then used somehow selective extraction to reduce sample complexity. So after dicing the sample, grinding samples under liquid nitrogen, which leads to a fine powder, which can then be nicely extracted. We have used 0.0 molar potassium chloride, and we mainly extracted the myofibrillar and sarcoplasmic fraction, and thus reduced the complexity of the proteome at this early step already, because we are not really interested in the insoluble or barely soluble proteins, or the proteins which are not that abundant. And with this, we then went to the tryptic digest and performed our bottom-up proteomics approach. The next step was then after the tryptic digest was the separation and high-resolution mass spec experiments. We have used several instruments, and among these instruments was also the ABSIX 5600 triple TOF instrument coupled to an Exigent Nano LC. And what you do on this instrument is that you then detect the exact mass of the precursor ion and then perform fragmentation experiments of as much precursor ions as possible. And I've already mentioned you can identify 30,000, maybe up to 50,000 peptides per run in these experiments. And under ideal conditions, you get really nice fragmentation spectra. And with these data, you can then identify your peptides and characterize the sequence of these peptides in combination with database searches. And keep in mind, the databases are not complete. Therefore, we, in addition, performed de novo sequencing to really try to close these gaps of these incomplete databases. And the main advantage is that you now really can do these database searches from all your data. So from this is really at least something like proteome-wide. You have at least 5,000 or maybe 10,000 peptides, which you can search in parallel, and you do not have to click from protein to protein on your BLAST website. So this is really huge advantage. And the data sets you obtain from these experiments can be quite complex, and they are often displayed in something like this here. This is a heat map where the retention time and the m over z values are given on the x and y axis. And every circle you can see on this heat map represents precursor mass of a peptide together with an MS-MS spectrum. And these data sets are then used for the database search. And this is exemplified here. You can do your database searches. This is only a tiny window of this heat map, so we have a retention time window of about 3 minutes and only a small stretch of the M over Z values. And all the circles are now colored in different colors. So we had a pork sample, so scrofa, and you see that most of the identified peptides are non-specific, or the peptides which are red are non-specific, and you also do see 2 blue peptides. These are the ones which are, at least according to the database we have used, are specific to scrofa. In this experiments we have used a FAST-R file that contained all sequence information of all relevant mammalian species that are relevant for food production or relevant for contamination of food, so to say. So the 2 blue ones are then the potential candidates that might be specific for pork. You can also see that databases are indeed not complete, because if you have a look at the 2 pink dots, these are specific, but they are not specific for scrofa, at least according to the database. And keep in mind, it was a pure scrofa, a pure pork sample, so these are false positive hits for specific peptides due to incomplete databases. And this is also underlined by the 3 white points, which are peptides that are not covered from the databases. So this is one of the points you really always need to keep in mind, that you cannot only rely on databases, you have to do additional validations. In our experiments, by using a soft extraction method, we reduced the complexity of our proteome, and we were able to identify 5,500 peptides from 500 different proteins using the ABCX triple TOF machine coupled to the Exigent Nano LC. And from the automated database search, we found 500 peptides which, according to the FAST-R file, are specific to pork in this case, and this nicely fits with the sequence identity one will find in mammalian species, which is around 90 to 95%. So 90 to 95% of the sequence is identical, and 5 to 10% roughly are sequence polymorphisms, and this nicely fits with the database search. So we then used this 500 peptides to start and develop our MRM method, and we had several critical parameters which we checked all the 500 peptides for. It was first the amino acid composition, so we excluded peptides that contain methionine or cysteine due to the risk that these amino acid side chains are oxidized, then you have a mass shift and you will end up in false negative results. We try to avoid post-translational modifications, and we had a look at peptide length. Peptides that are too short only give not enough MRM transitions, so we excluded these peptides, and peptides that are too long show distribution of charge, so you have then maybe a peptide species from this peptide which has charge state of 2 and maybe also a charge state of 3, so you then might see a loss or reduction of sensitivity, so we also really kicked out the very long peptides. We tried to reduce amino acids that are prone to modifications during processing, such as the Maillard reaction. And finally, we searched all candidate peptides against the UniProt KB database in a halfway automated fashion to be sure that these peptides are not in relevant plant proteins or in microorganisms, because you might have the problem if you think about, for instance, salami, which is a fermented meat product. You have lactobacilli in there which do the fermentation, and if you have the same peptide which is produced by the microorganisms, you will maybe see false positive results. So you also have to bear this in mind for your method development. I don't want to go too much into detail concerning MRM because this was already covered by Steve's talk. You all know that your parent ion is fragmented in the Q2 in the collision cell, and you then see the fragment ions, the daughter ions, and when you fragment peptides with collision-induced dissociation, you will in general see fragmentation along the amide bond of the peptide, and when the charge during this fragmentation is then located at the N-terminal site, you will see so-called B ion, and when it's located at the C-terminal site, you will see a Y ion. So you will normally then get a set of so-called B and Y ions, and from the most abundant ones, you then choose your MRM transitions for your method development. We have performed the method development using the MRM pilot software from AB Sykes, because one of the problems you have when you have 75 peptides which might be suitable for the MRM method, you will end up in something like 400, maybe 500 MRM transitions, and it is simply not feasible to manually optimize 500 MRM transitions. So we used the MRM pilot software, and we were really quite happy with the results. The first thing MRM pilot does is the validation of MRM transitions. So you have automated method generation where dwell time and cycle time and so on and so forth is taken into account. And you repeatedly measure your samples of interest and then see whether the MRM transition occurs or not. And you have some secondary experiments where product ion spectra are recorded, and you can then also from MRM transitions and the product ion spectra see whether this MRM transition is really from your peptide candidate. And from the validated MRM transitions, you have in the next step the iterative optimization of MRM. This is mainly collision energy. And finally, after this MRM pilot optimization, we then checked all potential biomarker peptides, and these were in our cases roughly 20. We had more, but we excluded all the other ones. We simply took the 20 most sensitive and most abundant ones, and we checked these 20 against all other species of interest. So for our pork samples, we checked them against horse, lamb, chicken, and beef, and vice versa for the horse samples, ending up in 5 to 10 peptides, which are really also specific on experimental basis, and we again only took the 5 to 10 most sensitive peptides. This is shown in the overview here, starting from the 5,500 peptides. After database search, identification of suitable peptides and MRM development, we ended up with the 5 to 10 most sensitive peptides. So we still have some more in the pipeline which are also nicely specific, but they are less sensitive, and this is the reason why we kicked them out of our method. And the next question for us was then, how many MRM transitions really do we need to be specific? It is in general the case that for small molecule analysis you need 2 MRM transitions, and when you really see that you have decreasing concentrations of your peptide in your samples, you will see that you might start with 5, 6, or 7 MRMs, and you will lose 1 MRM after another the lower the concentration of your analyte is. And the question is, how many MRMs really do we need to still be specific? And We did some in silico tests where we had artificial MRM transitions from a peptide from beef. And we searched these data against human database sets to have a look at how many false positive results we get. And when we have only 2 MRM transitions and a mass accuracy from 0.5 Dalton, we find in the human data set 25 human peptides that were false positive, and even when we increase mass accuracy, we still saw 9 human peptides which were positive and false positive. So obviously 2 MRMs are not sufficient. We did the same with 4 MRM transitions and searched them against all mammalian data sets, and with this approach, we still only find our peptides. So with 4 MRMs, we're really highly specific against all mammalian peptides which are in databases. So this really was a nice result. But in addition to the MRM transition, we also have retention time and signal ratio as additional confirmation. So we ended up with 3 MRM transitions together with retention time matching signal ratios and the experimental validation of your species specificity. These were our main data where we say this is still specific and this is what we need to be sure that we see this species really in our samples. And this is the overview of the identified biomarker peptides. And please excuse that some of the sequences is somehow crashed. So if you download the paper, you will see the complete sequences. This is just for an overview that we finally identified 12 biomarker peptides for our initial study. And the next question was then, after having an idea how many MRMs we really do need to be specific, is there any further possibility to increase sensitivity? And Steve already mentioned that micro-LC really can do a job on this, and we had the similar results that using micro-LC we were able to increase sensitivity by a factor 3 to 5 roughly compared to conventional LC, and we also were able to have a faster chromatography. So we reduced chromatographic time from something around 40 minutes to something like 15 minutes, so roughly factor 3. And I've seen that some of you really are interested in increasing throughput, therefore micro-LC might really also be interesting for you with this respect. And these are this is the overview of the chromatogram of all our biomarker peptides. For clarity, the most intense MRM of every peptide is given in red, and the second intense is always given in blue, and the third is always given in green. You can also see that there are some unspecific signals which are marked by asterisk, and you have to bear in mind that you have highly complex samples which contain 10,000s of peptides, and what you see is then that you always will have some unspecific single MRM transitions which pop up, and this is simply due to the fact that you have so complex samples. And when you zoom in, this is the zooming in, or the retention time window of the 3 most intense and most sensitive peptides. It's a 2-minute window, and the complete the complete runtime was about 15 minutes. So this is something like 15% of the complete run. You see that here specificity is really nice. You see the first marker, which is specific for pork and horse, is nicely seen in both species, while it is not observed in beef, and even the second marker in the middle, which is specific for pork. You can see that in the horse sample there are single MRM transitions occurring, but you have always you have never the retention time match, and you have never the correct signal ratios, and in nearly all cases you only have a single MRM transition which occurs from one single peptide, which is would be a false positive hit, and only one single MRM transition which comes from these peptides. So specificity really is very good, and this is seen also for the other species, for chicken and lamb, where you also do see sometimes these unspecific single signals, but when you really bear in mind that you need 3 MRM transitions and retention time match, specificity is really convincing, at least from our point of view. The next step I briefly want to introduce to you is MRM cube. You know how MRM works, and the thing about MRM cube is that you, from your daughter ion, so you have in MRM you have a parent ion which then is fragmented to daughter ions, and in MRM cube you then have, so to say, the granddaughter ion. So from this Y fragment ion you have an additional secondary MRM transition and fragmentation in the linear ion trap, which then leads to this secondary fragment. And this really helps increasing sensitivity and also specificity, which is shown here. This is the chromatogram at the limit of detection of the respective peptides, and you can see that in the upper row you only have one MRM transition, and when you do the MRM cube experiment, you really see a massive increase in signal to noise and really enhanced specificity. So you will be able, when you at this stage perform MRM cube, to nicely really detect your species, and this is also shown for the horse marker, where you also have this nice MRM cube. So we were really surprised to see how good MRM cube really works. And with this, I'd like to give you some future perspectives. I hope that I can show you that mass spec really has great potential to be used in future routine analysis of meat speciation. And we are currently working on the analysis of processed and highly processed samples, so fried and cooked meat, corned beef, and so on and so forth. And we do see that when we optimize the extraction and use more harsh conditions, we are able to extract our proteins of interest and to still generate our biomarker peptides. And we were quite happy to see that we do not lose that much sensitivity even in the highly processed samples. So we are now at the stage of writing our publication, and I really hope to that we can provide you with these data in the near future. And in addition, we are working on the multi-species method, so to integrate further species in the current method, 8 to 10 species roughly, so that you have one method which you can use and you can really in parallel analyze and detect several species in one run. And with this, I'd like to acknowledge ADSciX and especially Steve Locke and Dietmar Weidlich and Jörg Dojan for all the fruitful discussions, and also the opportunity to do some measurements on the triple TOF instruments and the 6500 Q-traps in Darmstadt. And I'd also like to acknowledge Naturland and Archhof Bühning, because they provided us with several pork samples to assess breed specificity. And I'd like to thank all of you for your attention. Thank you very much.
Thank you, Jens. A very, very interesting presentation and how to see how mass spectrometry can be used in this sort of very selective and sensitive a way to detect those protein markers specifically for individual species. Really interesting presentation, and I hope everybody on the line and the WebEx enjoyed the content. We do have some questions that have come in through the question and answer section. So if there are people who have any burning questions, please do add any further questions. But I think we can ask a couple of questions, Jens and Steve, just to clarify a couple of points. One question that's come through is said, does the fat content of the sample have any negative impact on your results using this type of approach, this method?
If you have really fatty samples, then the main impact really is that your protein content is reduced, and by this, of course, sensitivity is affected. So in our initial study, we have used high quality meat, and if you have really high content of fat, it's simply the case that you will have lower protein contents, but we do not have the impression that the extraction or the downstream work is really affected. It's mainly the reduction of analytes.
Interesting, yeah. It's sort of, as you say, from what you've shown, the sensitivity is not really affected by that. So, yeah, quite clear. We have another question. I think you touched on this just in the final commentary in terms of conclusions on future work. Would this method work for both raw meat samples as well as cooked or processed food samples?
Yep, it will. So we were really surprised. We thought about, okay, we will see loss of sensitivity, but it isn't that much. It isn't even factor 2 or something like this. It is a slight reduction of sensitivity. You see more background, mainly I think because you extract more peptides and you might have Maillard products, and we really had deep fried meat products, but it still nicely works. We were surprised by ourselves, so yeah, it will work.
Okay, good, thanks. Another question is, do you think this method would work with other types of food products, such as, say, for example, milk, so milk speciation and authenticity?
Yes, so of course the approach in general will work. I mean, it's similar when it comes to, for instance, allergen detection. You can apply this form of identification of biomarker detection in principle for all kinds of food. And we do similar things for the detection of allergens, for instance. The first thing you, of course, have to do is to identify the biomarker peptides in your milk matrix. And if you have specific biomarker peptides, milk would absolutely work. I'm convinced. Yeah.
Okay, and I suppose this next question relates to that as well. Do you see any other food fraud trends coming? Obviously, we saw the big scandal with the horse meat in 2013. Do you see any other food trends or food fraud that we may actually see this potential use of mass spectrometry will be applied to?
With respect to food fraud, I think that meat authentication is one of the major points, because the profit margin is, I think, the highest for meat authentication. I have seen that there is a conference in 1 month in Belfast, the ASSET 2014 conference, and there's something around authentication of spices and also of plants. So in principle, this is of course also achievable. They use other methods, but you can of course use MS, and not really for the authentication, but for the species identification. Allergen detection is similar to that. So if you want to know whether you have hazelnut in your chocolate, this is nothing else than species identification of hazelnut. It's not really authentication, but it's protection of allergenic consumers, so to say.
Absolutely, yeah. Peanut allergies are a big issue, aren't they, with regards to food? But as you point out, the actual amount of money that the industry and people who are basically defrauding the consumer is quite considerable. As you say, meat is so expensive. I think it is a big topic to. There's a lot of money to be made by people defrauding consumers, so that's a good point. Yeah. And I suppose this final question, this is the $66 million question. Do you think that there is a potential for mass spec to replace methods like ELISA for routine meat speciation testing? I mean, the presentation certainly shows the capability, but I suppose it's a $66 million question.
Yeah. So actually, I mean, future will show, but I think that the distribution of mass spec instruments, from my point of view, is really increasing. So there are nowadays many labs having a mass spec instrument, and the nice thing about this method is that once these methods are published, you do not to do all these laborious proteomics approaches and so on and so forth. You simply have to take your biomarker peptides, and if it's a decent study, you can use these data and implement it on your mass spec platform, even if you have no idea of protein and proteomics and protein chemistry, because if you degrade your protein to peptides, they behave similar to small molecules. And I'm quite convinced that mass spec will have its place, and I do not dare to tell whether ELISA really will go down, but I think that mass spec really has great potential and has a good place for food authentication if you think about multiplexing and so on. So I think in the future mass spec will be relevant.
Yeah, I think that's a good point to make. Maybe one day in the future we might see a mass spectrometer in a slaughterhouse, for example, or a production line. Who knows? Online testing. Yeah. Okay. Super. Thank you very much for your answers and questions. And again, thank you very much for your presentation. Very interesting talk. We've just got a final couple of questions to ask the audience, and while we wrap up, the presentation has been recorded and it will be available on the AB SciEx website, and all the participants will receive a link to the presentation. Don't forget also the files are for download, the paper that Jens has written, and also a couple of application notes from AB SciEx, which sort of talk to this use of mass spectrometry for protein-based detection. And hopefully everybody has a pleasant evening, pleasant day, and thank you again, Jens, for your time during the presentation today. Thank you, everybody. Bye-bye.